Thank you for coming. For the neighbours who are here, this annual Dorcus Cummings Lecture has been going since 1978. It's usually held during the Cold Spring Harbor Symposium on quantitative biology. And this meeting, which is going on now, it started on Tuesday and finishes tomorrow evening. And the symposium, as I said, has been going since 1933, and the topic changes each year, and we have it on a hot topic in biology or medicine or the life sciences. And this year it's on brains and behavior, and there's been some fascinating, really, truly amazing science that's been presented by some of the world's top neuroscientists at this meeting, and will be tomorrow as well. It has been a tradition at Cold Spring Harbor Laboratory, going right back over 100 years, to have interactions with the community, and particularly public lectures. We have a whole series of public lectures during the year, and also movies and concerts in this auditorium. But this is a named lecture, the Dorcus Cummings Lecture. As I said, it started in 1978, and by the Cummings family, Robert Cummings, in honor of Dorcus Cummings, and we're very happy to have Robert Cummings and his wife Poppy here, so thank you for coming. And we're also outside, there's a poster of all of the speakers who have spoken at the Dorcus Cummings Lecture, given this lecture over the years since 1978, and it's really an incredibly impressive list of scientists who, many of whom have won the Nobel Prize and many other very prominent awards. In selecting a speaker, we try to have someone who can actually speak science to the public. So for the scientists in the room, you're going to see how Reid performs. But for the lay public, I hope that, well, I know that this is going to be understandable, because Reid is a well-known public speaker, has given TED Talks, and also has an engaging topic. And so the purpose of the topic of this meeting is on brains and behavior, and Reid is a professor at Virginia, the director of the Virginia Tech Carilion Institute at Virginia Tech, and also a professor of physics. And he works on computational neuroscience, and he's going to give a lecture tonight connecting the mind and the brain in a computational age. So it is my great pleasure to introduce Reid Montague.
Okay, I'm a little nervous. The main reason is after I was invited, I thought this was hey, could you talk to the donors and the people in the public here? I thought it was sort of an informal thing the next day, and then I looked it up, and then I got this email saying that we all remember with awe and admiration the inspiring lecture given by Francis Crick. Okay, Francis Crick, along with other people in the audience, discovered, you know, the tape on which life was written, and it was a beautiful molecule. So I'm going to manage expectations here. I am no Francis Crick, or Jack Kennedy for that matter. Okay, I also have another position that I often fail to advertise, that I was told to advertise. I'm a PI at the Wellcome Center for Human Imaging, Human Neuroimaging at University College London. Okay, so I'm going to be a bit interactive here. I'd like to see a show of hands of people who either own or have driven or ridden in a Tesla. Okay, all right. How about the owners? How about the owners? Okay. Yeah. There was an age distribution there, by the way. Okay, and of the people that have the Tesla, what do you think about the automatic driving capacity? It's sort of amazing. It drives itself, right? And you can let it. No, I see a lot.
We do a better job, though, as human beings, which I hope will inform your talk.
Okay, I'm gonna start the talk by breaking one of my 2 rules of talks that was taught to me by Jerry Adelman, who was one of my sort of 2 postdoctoral mentors. And this is by my other postdoctoral mentor, Terry Sinofsky, who is, by any measure, is a pioneer in computational neuroscience and bringing it from the back roads into a prominent position in neuroscience that it plays today. And we heard, not everyone that's here from the public is privy to what was said. I'm going to let people read this as I'm talking. First of all, my wife objected to the data are the new oil. She works for Oracle Corporation. She said, you can use the word is. It's a usage thing. And of course, if you're a scientist, you're very aware of the difference between a datum and data. Terry's a scientist, so he opted for this. I'm going to read this to you, though, and then I'll zip through the rest. It's often said not too long ago that computer vision was not able to compete with the visual abilities of a 1-year-old. And that was a defense, as it were, against we've gotten nowhere with computers and computational systems and neural networks. Okay? This is not true anymore. This is why I ask you about the Tesla, because I have friends that let their Tesla drive them to work an hour on Highway 81 and basically never touch the wheel. Okay, maybe they're foolhardy, but they're still my friends and not dead yet. So something's working. It's no longer true. Computers can now recognize objects and images about as well as you can, and there are cars on the road that drive themselves more safely than a 16-year-old, a low bar. Moreover, computers have not been told how to drive or how to see, but have learned from experience, following a path that nature took a million years ago. I don't know about the million years. Millions of years might have been longer. What is fueling these advances is gushers of data. Data are the new oil. Learning algorithms are refineries that extract information from raw data. Information can be used to create knowledge. Knowledge underlies understanding, and understanding leads to wisdom. Now that's quite the rhetorical flourish from Terry. So I buy that. And I'm going to give some examples of this. And I'm going to say that neural network approaches to hard problems now, which are also being poo-pooed as, oh, well, they're not really playing chess the way people play. That used to be the defense 30 years ago. Okay, you've got this mouse, it runs around a maze, and it falls over on its side, and it's the size of a vacuum cleaner, and it takes every supercomputer in the world to keep it running for 45 seconds. But you could never do something as hard as chess. And chess is gone. The game of Go is gone. Every Atari video game is gone. Computers completely crush human beings on these games, trained in really, really simple ways. And the ways they're trained are the same way that your nervous system trains you to learn about objects in the world. They're trained on something called reinforcement learning systems, about as simple as they could be. And that's what we're going to focus on here. I actually think the wisdom bit is a little bit of a far reach for Terry at the end of that, so we're just going to stick to the information, knowledge, and understanding part. I won't be able to comport any wisdom on you today. Okay, so what's common across these creatures here? Some of these are creatures. I didn't name them. Honeybee, rodent, zebrafinch, one of my 4 girls. AlphaGo, this is the game of Go. For those of you who don't know anything about Go, it's a very complicated game played on a 19 by 19 board. It's sort of like Othello on steroids. You capture territory, and there are lots of moves. It's a very complicated game. It has a gigantic state space, something on the order of 10 to the 150th power. Okay? There are about, if I round up, there are about 10 to the 80th particles in the universe. So 10 to the 150th is really a big number. The branch factor in Go, meaning the sort of average number of moves from any one board position available to you, is on the order of 250. Chess is on the order of, I can't actually find it. I'm sure there's a chess expert in here. But as far as I can tell, chess is between 30 and 40 branch factor. So any given board position, you have 30 to 40 moves legal available to you. Okay, these are really hard problems. And that's why before Deep Blue beat Gary Kasparov in 1997, IBM's effort at making a chess playing program, people thought it couldn't be done. They thought it was just too hard. I don't think that proves anything particular about Deep Blue. I think it says something very deep about Gary Kasparov. Gary Kasparov played basically the history of chess, turbocharged in a supercomputer, built-in grandmaster, knowledge, and could do 200 million look-aheads a second. He plays it to basically a tie, and he can have a political opinion and think about the cheese sandwich he's going to eat after the match. And of course, Deep Blue can't do anything like that. It's not adaptive. Okay, so human beings are still pretty cool, but they've been crushed by this, okay? What do they have in common? And the answer is they have algorithms in common. They have learning procedures in common. They have software that's in common. The heart of the programs that play backgammon. This is called breakout. When you hit a little ping pong ball, it knocks these pieces out. And this is called go. The heart of that is something called evaluation function. And it's just what you think it is, okay? You put a bunch of data in front of a neural network. The neural network rearranges the data and adjusts a valuation function, which for our purposes is basically a lookup table. I'm in board state 3. What are the values of all the boards, all the 250 moves that I could make now? I'm going to assign a number to every one of them. Basically a big, giant lookup table. Okay. Now that doesn't seem very human-like. It doesn't seem to be the way that you feel like you think. But the way you think and their instruments that build the way you think are not what you expect at all. Okay? And what I'm going to tell you tonight, I'm going to tell you two things. I'm going to tell you the sense in which computational approaches to problems, to behavioral problems, and even understanding neural circuits is an alternative way of looking at the data that's been presented this weekend. And the data now are amazing. The techniques available to neurobiologists now are really, they blow my mind. And I've been in the field, 25 years. And I would never have imagined we'd be where we are now, asking the kind of questions we can ask. It's not enough just to understand the parts, because biology's got a lot of variability in it. We kind of want to know what they're doing. And frankly, learning evaluation functions like these programs do here is so important. And prediction, which is the heart of how these evaluation functions are learned, prediction is so important that I think it's probably been rediscovered by evolution over and over again. Okay? It's thought that eyes evolved anywhere from sort of 40 to 50 separate times. Creatures that use vision to survive have developed all kinds of strategies, and we think of this as independent evolution of eyes. Biology has discovered eyes over and over again, all kinds of different eyes. Insect eyes are very different than the eyes in your head, for example. Okay? I think the same could be said for learning algorithms, that you should expect these learning algorithms not to be implemented in exactly the same way across phyla, but there to be large patterns that you could maybe detect, and I'm going to talk about one of those today. And then I'm going to segue to an area that I think is quite forward-looking, and it's just now booting up, that we're going to call computational psychiatry. Okay? So biology discovered structural and algorithmic motifs, is what I'm arguing. So the thing in your head and the thing in these creatures' heads that does the reinforcement learning, we're going to fill that out in just a second, are neurons that look like this. So this isn't really quite I couldn't find an analogous picture here. This is a dopamine axon coming up from an area in the brainstem of a rat, projecting to the prefrontal cortex or the striatum, and this is an arbor of 2 axons. Okay. That's one neuron. There's one neuron hooked to this thing down here. It creates electrical activity. Little impulses run up here. They divide at each of the branch points, and they go out and they communicate whatever it is they're communicating to this bush here. This is gigantic. This is in the subesophageal ganglion of the bee, the honeybee. This is a neuron that I know and love called VUM-MX1. I won't tell you why it's called that. This is from the work of Randolph Menzel and the late Martin Hammer. And this projects out throughout the bee brain, but it directs a certain kind of reward learning with the connections that are up in this part of the brain here and here. Okay? You have analogous systems in the human being coming from the substantia nigra and the ventral tegmental area. These are clusters of dopamine neurons in your brainstem that, like this rat neuron here, project through large expanses of the cortical mantle into an area called the basal ganglia. These are the neurons that you lose when you get Parkinson's disease. If you show up in front of a doctor with symptoms of Parkinson's disease, you've probably already lost 70 to 75% of those neurons. Now, that's a kind of cell biology, molecular biology, early trauma insult question of how is it that they started dying. But once they start dying, then your brain doesn't know how to value actions and sequences of actions correctly. And I guess I would say that it's both a motor problem and a valuation problem. And we can come back to that if you're interested. This is the only source of dopamine, other than in your pituitary gland, where there's a little projection from your hypothalamus to the pituitary gland. This is the only source of dopamine to your brain. If you lose these neurons, that's it. And in a sense, life isn't worth living at that point. You act as though there are no rewarding consequences to any of your thoughts or your actions. It's a really important nexus of neurons there. They're not the only neurons that do stuff like that. There are neurons down here that have the same sort of general projection patterns that deliver the chemical serotonin. Serotonin and dopamine, if you just looked at the prescriptions written in America, the number of prescriptions written for treating or perturbing your serotonin system for depression, SSRIs, for example, or touching your dopamine system is probably 70 million people. Okay, so if you were going for the two biggies, you might start there. But there are others too, acetylcholine, various peptides, etc. And this is the bird brain. This is the zebrafinch. This is a cartoon, obviously. It also has something analogous to the ventral tegmental area. This is a dopamine projection, and it controls the way the bird learns to mimic the song of a male. Okay? There's a theme here. The theme is, I'm going to outline here in just a second. So this is a diagram from a book on computational neuroscience from 1992, when it was barely an embryo. I think it was in utero, computational neuroscience in 92, written by Pat Churchland and Terry Sinofsky. I was in the lab at the time at the Salk Institute. And this is the classic scales diagram. And this is the classic way that people sort of decompose the problem of understanding the nervous system. Scales in space and time. Completely reasonable. Things at different scales take place on different at different time scales, spatial scales. They require different kinds of forces, etc. Okay? In 1988, they had published a paper right before the book where they plotted, let me explain the back one, then I'll show you this one here. This is space here on the log scale. So this is 1, 2, 3, 4, 5, 6, 7. This is 8 orders of magnitude from the, sort of the size of the brain down to a synaptic connection between two neurons. And these are time scales here from milliseconds up to months. Okay? And what's plotted here are the various techniques we have in 2014 for studying this space-time decomposition of the nervous system. Okay, well that's reasonable. That's a reasonable way to take on the problem. It's a really hard problem. You hear that in everybody's talk today, despite the fantastic techniques. But in 1988, this is what it looked like. One big gap got filled here by functional magnetic resonance imaging, and another gap was filled by optogenetics, which actually continues to expand the scales at which it's been active all the way up. I would say that optogenetics probably goes up to the size of the whole brain now, given what I've seen in the zebrafish, and the techniques are really explosive now. Okay, that's not the only way to decompose a problem, though. Another way is to take computational primitives, as it were. Paul Glimcher talked about a computational primitive earlier, I guess 2 days ago in this symposium. But I'm going to talk to you about prediction learning. Prediction learning is just what you think it is. In a sense, biology is all about prediction, right? A creature is challenged by an environmental challenge. Something inside the creature rearranges, and then right after that, it's better at responding to a similar or generalized challenge, okay? Learning. Bacteria do that. Birds do that. People do that. Herds of people do that. People interacting with machines do that. It's a principle that you could go looking for at many scales. Okay, so we're going to use that to go looking for substrates, physical substrates of prediction learning. In particular, we're interested in how you learn about things that are rewarding to you, how you learn about food, water, sex, and salt, things that keep you alive and things that keep the species propagating. If you don't do that, it doesn't matter whether you drive a Tesla. You know. All right. So neurons and synapses live in this range, and so we're going to ask the question, how can synaptic plasticity, changes in synaptic strengths between neurons, support the kind of learning? And I'm going to dial back to a big idea had by a now very famous, I guess I'd call him a psychologist, Donald Hebb. About the same time, Jerzy Konorski, a Polish neuroscientist who was somehow overlooked for various kinds of political reasons, published the same idea the year before. This is in 1949. Hebb published the following idea. It was based on the following instinct. Correlations and associations in the world seem to change what goes on in your head. You see the red bird flying overhead, and the next thing you know, you see the poop on your picnic table. Every time the red bird comes over, he poops on your table. There's something wrong with this red bird. He likes to poop on the table, okay? Eventually, you know, if you're learning at the right rate, you learn the red bird comes, I got to go get the sponge, I'm going to have to clean the poop off the table. Those associations, those correlations, must be changing something in your brain, so Hebb upgraded that to a physiological hypothesis. And he said, suppose that things that look like correlations in the world end up being increased synaptic connectivity between two neurons that correlate with one another. Okay? the fire together, wire together idea. So the bird and its redness and the poop are all happening sort of at the same time. There are neurons that are active during that. They're co-active, and that co-activity, action potentials or electrical spikes in neuron A paired with electrical activity in neuron B causes this connection to change its efficiency. Those change efficiencies are like the numbers that I was talking about in the valuation function for those games earlier. They learn those associations. Good idea. And it was brilliantly simple, and it made lots of sense, and it's wrong. This is a paper in 1984 from Eric Kendall, Tom Abrams, Bob Hawkins, and Tom Carew. It was a famous paper for me because it came out the year after I graduated from college, and it was testing just that idea. And it was testing it on a sea snail that had a little gill withdrawal reflex that it protected itself by. And that gill withdrawal reflex is, you stab the sea snail's tail, it sucks its gill in, and then it puts it back out and sucks it in. That reflex could be sensitized, made more sensitive and quick, or it could be habituated. Okay? And they were interested in using this to see if they could look for the cellular basis of that simple learning in that simple creature. And the answer is we've tested directly Hebb's hypothesis, and it's neither necessary nor sufficient to account for what they see. And they noticed that impulse activity in another neuron that was paired on what's called the presynaptic part, the sort of the input end of the synapse, was required. And that stuck in my head. I showed up at the Salk Institute in 1990 as a postdoc, as a kind of converted applied mathematician, having gone through the rigors of a neurobiology course at Woods Hole and come to actually something here. And Terry Sinofsky, the head of the lab, it was an oddball lab in the sense that Howard Hughes supported it back in, I want to say, 1989 or something like that. Okay? And I met this guy, Peter, so we were litter mates in his little hovel, and we were sort of assigned a problem. I want you to study diffuse ascending systems and figure out what they do. Okay, well, what information do diffuse ascending systems encode? So we were just talking about these earlier. These are the systems of neurons with their cell bodies in the brainstem that send projections throughout the brain. What are they doing? Why is there such an elaboration of that? What is it about? What kinds of synaptic changes could they direct? And that's what we went after first. So the first thing we did was we decided that we needed to change Hebb's rule. Okay? And the reason we decided that was a long series of research, really starting in the 1950s, not just with Richard Bellman, with a guy called Samuels, who made an automatic checkers playing program and came up with a learning procedure which was super simple, not quite right, just a hair off what he could have guessed, but didn't quite, called the Bucket Brigade. And it played checkers, and it could beat humans at checkers. Okay, checkers is a pretty simple game. There was another guy, Richard Bellman, I think he was at Rand Corporation at the time, who was working on something called dynamic programming, which is dynamic programming. He named it that because he thought that was a happy name. He couldn't see any negative connotations to the term dynamic programming. But dynamic programming takes on the following problem. You have a game you're playing, so you make a choice, and then you have a set of other choices. You make one of those, and it keeps branching, okay? And you run all the way out to the horizon of the game to whether you lose or win. And it's a principled way to back that process up and learn optimally how to play the game, how to tie together sequences of actions that lead to the most wins. The problem with dynamic programming is it's kind of impossible to do on anything but trivial problems in its native form. Okay? Most games even simple games are too hard to learn. Rich Sutton and Andy Bartow, through the 80s, came up with an idea that ended up being something called the method of temporal differences, and I'm going to show you that in a second, mainly through waving my hands. The method of temporal differences. And a guy called Christopher Watkins at Cambridge University wrote a thesis in 1989, which is one of these singular breakthrough theses where he fused these two ideas. And he was working with Peter Dayan, who had just come to the Salk with me, on making a proof of the convergence of this method. And he called it Q-learning. So it blended together the best aspects of dynamic programming and the best aspects of sort of animal learning. Rich Sutton based his method on psychology of animal learning. Even though he ended up with a PhD in computer science, he had been a psychology undergrad at Stanford and had all these ideas about animal learning in his head. Okay. If that's not a good soporific, I don't know what is. All right. So we took Q learning. It would be worse if I showed you the methods. So in one sentence, what it is it's a method to learn to maximize your returns over sequences of actions that are possibly long-term. Okay? Let me give you one example. Suppose I took a table and I set it up here, and I put a little cage in the middle of the table, and I put a bug in the cage, and the cage has a little door in it, but the door is closed. The bug just wanders around inside the cage. And now I open the door, and the bug wanders off, and it wanders along the table, and it falls off the table, and it bumps its head. Okay? What made it bump its head? Well, it should assign a lot of negative credit to that last step off that big cliff. You see a big cliff, stop, turn around. But the key point was the moment he stepped through the door. The moment he stepped through the door, every path led to Rome. Every path led to a negative outcome. What Q learning does, incrementally, is say, how do I assign credit to that point for this outcome that's temporally distal? Okay? It's called the temporal credit assignment problem. So anyway, so I met them, and they taught me what they were doing with Q learning, and we all, who can remember, I don't remember how this went, came up with the idea that that was a perfect way to sidestep Eric Kandel's problem from that 1984 paper. He's got something on a presynaptic terminal. It doesn't look like Hebb rule, but there's something that depends on it, and so we changed Hebb rules. We present a local learning rule in which Hebbian learning, correlational learning, correlations in the world translating into correlations in your head, storing a parameter, is contingent on an incorrect prediction of a reinforcement signal, a prediction error signal. The system's constantly making, some system is making a prediction of how much reward you're going to get and computing differences between the actual reward you get and the prediction, and that's feeding into synapses. This is what that would look like in a schematic way. This is a bit of a crawl. I get that. This is a bit of a crawl. We're crawling up. Now the idea is action potentials here correlating with action potentials here need this other input, and this input controls the sign of the synaptic change. That's small. It makes a big difference. Now instead of storing correlations in your head that are consistent out in the world, you store predictions, and you can use a prediction to decide what to do next. Okay? That's the idea. Let me show you one slightly technical slide. So here's the way it would look. Here's the way it would look for a mouse running in a maze. And let's discretize the maze. One point, next point, next point, next point. You go right, you get a rock, you go left, you get food. Okay? What this algorithm does, and I'm not going to explain the equation, is it has a goal of learning. And the goal of learning is to learn the value of the state at time t. And the value of the state at time t is going to be the average reward you can expect from that state to the future. And we're not going to care how you got in that state. Okay? We don't care how you got in any state. It's going to be a history-dependent, independent process, but that's the goal of learning, to estimate the value of some state, some board state in Go or some board state in chess is going to be the expected value of the rewards you can expect in the future, and you can tie those rewards to either winning the game or losing the game. When you write down that equation that way, when you say that's the goal of learning here, it has a natural error term that falls out of it. I'm not going to go through that. This is what we thought the dopamine signal was. And what is this error term? This is called the temporal difference error term. And what it is it's a difference in time between the value of the next state that you're about to engender, the value of the next state in time, value of the current state, and anything unexpected that happens to you. It's a successive prediction model. So it goes like this. I predict I'm going to get 3 units of reward next time. That prediction, plus whether or not I get 3 units of reward, can change my error term. So 2 things can change the error. Your predictions change through time, or the reward expected is different than what you expected. Both of those things can cause the signal to go high, things were better than expected, or low, things were worse than expected. There are neurons in your brain that act precisely like that. And you can show, and I think I indicated, algorithms that use this sort of setup to learn to play things can beat every player who's played the game of Go in the world. So you have to know a little bit about the history of this Go game. They tuned the Go game initially using grandmaster input. And they tuned it so that they could play the Chinese champion and the South Korean champion, and it won, and it kind of devastated the Go world. Then they made another program called AlphaGo Zero, which trained on no human input at all, and trains up to Criterion in about 10 hours, and trains to Supermaster level in about 30 hours, and it beats AlphaGo 100-0. It basically beats the entire history of Go. Okay, so now the complaint is now, well, people don't really learn like that. They had these intuitive algorithm stuff. Maybe, or maybe hard problems like that are just like lookup. You have neurons in your head that emit signals exactly analogous to the signals they use to criticize the moves in those games and learn that value function. Okay? I think one way of thinking about that is that biology stumbled into this algorithm, right? Biology stumbled into this algorithm multiple times. And I showed you what it did. It ends up with structural and computational motifs that seem to be very analogous when you look at bee brains and mammalian brains and the brains that play Go. So take-home point number one. We have in our head substrates of prediction and reward that rival the way neural networks now beat what used to be thought of as fantastically difficult problems. I'm not going to go through this. That kind of activity, let me go back, shows up in the activity of dopamine neurons in your brain. So let me just point out something here. So this is time on the x-axis, and this is average firing rate on the y-axis. This is a histogram here. But each one of these traces is a single trial. We're listening to a dopamine neuron while an animal is doing a classical conditioning task. There's a cue that comes on, a picture, goes off, wait a while, squirt juice in the animal's mouth. When you squirt juice in a naive animal's mouth, the dopamine neurons go pop, 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 pop. They pop along. These are little electrical impulses in the neurons. If you keep doing that, pairing a cue followed by a reward, and you keep the time consistent, this is what happens. Now all of a sudden, when you turn the light on, the neurons, brrr, they give a burst at the early predicting light. The light predicts the juice, right? And now when the juice is delivered, nothing happens. So two things have happened. The response to the juice has gone away, and the response to the early light has grown. This is exactly like a temporal difference prediction error. What's going on silently, I've shown you right here. What's going on silently is from the time of the cue all the way out to the time of the reward, you've learned a value function. You've learned a set of numbers that say how much juice you can and cannot expect to get, how much reward you can expect to get through those intervening times. That's a simple little algorithm, but it's very powerful when you hook it to a body and you put a bunch of other constraints and systems in there. So that's a recording from a dopamine neuron. That gets accounted for by the model I've been talking to you about. It's a causal mechanism. It only goes one way through time. Okay? It doesn't absorb information if the reward precedes the cute things that follow it. It only goes one way through time. You have to really work hard to make systems learn backward in time. Okay? So causality is built into biology in terms of learning in a very profound and deep way. All right? But it wasn't until 2005, these were based on the data of Wolfram Schultz, the empirical data of Wolfram Schultz, that a proper quantitative test of this got done in a beautiful paper by Hannah Baer and Paul Glemscher. Is Paul still here? No. Okay. He won't believe I said something good about him. So this paper made me feel really good that I wasn't wasting my time and we could sort of move on to other aspects of it. Just to indicate, bees have systems like this. This is a little neural network model in a bee. Several papers we did on that to connect this to decision making. Birdsong learning, same sort of thing. It has dopamine neurons that teach a bird to copy a template laid down by the male bird that's near it. They listen to it, they use that as an internal goal, and they train up their vocalizations after listening to a male bird for a year. Bees have it, birds have it, people have it. It's a theme, okay? When you go and test it in human beings, and I won't go through it, you see sort of the typical pattern. There's a sensory cue that comes on. There's a reward, literally squirting juice in people now, using functional magnetic resonance imaging. You can put people in a scanner and do the monkey conditioning experiment on them. Initially, you see responses early on to the reward. After training, these disappear. and they move back to the predictive sensory cue. And we're at the terminal place here. Now, this kind of imaging won't tell you that it's dopamine. It'll say that metabolic demand blood flow changes in these regions are following this scheme. These are the people that did those first experiments, Greg Burns, Sam McClure, and a separate group in London led by John O'Doherty. All right, you can even go and make recordings of dopamine directly in human brains. This is a group of people that I've worked with over the last 8 years, Ken Kashida, Rosalind Moran, and Paul Phillips. This is people being implanted with deep brain stimulating electrodes for either Parkinson's disease or essential tremors. We've designed a new way to extract dopamine and serotonin measurements off a small carbon fiber. This is a human hair here for scale. You can drop this down into a human brain and have somebody play a betting game. Make a bet. You're endowed with $100. You make a bet. Market fluctuates up. You win. Relative fractional change in the market times your bet. It fluctuates down. You lose. Relative fractional change in the market times your bet. When you do that, you can see that dopamine produces prediction errors, and it actually tracks the market quite well. Here's a market trace here in blue. Okay, that's the market. That's the value of the market. And these are slow changes in dopamine. So the fast changes, which I was showing you earlier, also are seen. But this slow change shows that sort of average dopamine levels in this person's brain were following the market. Market crashes. The person loses tons of money, but the dopamine signal pulled out. Okay, so the person playing the game lost 20% of his initial. He ended up with $80. Okay? The dopamine signal acted like it knew how to track the market and knew like how to pull out of the market. So we made a little artificial agent network that used the dopamine signal from this person's brain, and we asked, if you were using that dopamine signal, what kind of decisions could you make? And we let it do the betting. So we took the dopamine signal from this human brain, and we bet all in when the last 5 seconds the dopamine was sloping up. And we went all out when the dopamine signal was going down, and we showed that we made 75% over our $100. So they ended up with $175. The network ended up with more money than, at the time, 350 people had played the game. So that's an interesting finding. By doing it in a human, it's an interesting finding that this valuation signal in their head is somehow dissociated from their ability to get it out of their finger, get it all the way out to their finger. So that immediately starts saying, well, maybe the valuation's intact, and there's something wrong with coupling to movement. Okay? You might start sorting patients on that. So what we're hoping to do is move this method from this sort of specialized carbon fiber to the kind of standard electrodes that they use and implant in the brain so that we can make continual measures of this and really find out how we might intervene and see what the drugs and the stimulation paradigms do. All right. So what, right? I mean, okay, blah, blah, blah. So what? So these are all supposed to be rewarding things except, and unless you're a drug addict, this is drugs. I guess that's cocaine. Krispy Kreme, I grew up in Georgia, so Krispy Kreme was a big part of my history. Is there Krispy Kreme in New York? Is there a lot of Krispy Kreme in New York? No? All right, well, anyway. Why are these things valuable to you? Well, they're valuable to you because systems assign value to it, with learning. Okay, so let's do a thought experiment. The thought experiment's this. It's a little diabolical. Okay, let's imagine that every night, okay, this is a little sick, actually. So let's imagine that every night I sneak into your bedroom, not every night, on random nights, I sneak into your bedroom, I inject you with heroin. Okay? I use a really tiny needle. You can't feel it. There's no evidence of it the next day. You know, there are a lot of mosquitoes out. It's summer. I'm really, really clever. And I put it on random time schedule. 2 months later, are you addicted to heroin? Okay, your body's physiologically dependent on heroin. You feel like you know what. You're showing up at the doctor because I'll stop doing it in 2 months, okay? I'm going to stop. How are you going to feel? You're going to have diarrhea and a headache, and your body is withdrawing from heroin. But are you addicted to heroin? And the answer is you are not addicted to heroin because there are no cues. I've arranged it so that there are no cues that your nervous system has learned on that you can use to commandeer your behavior, that come into your behavior and make you reorganize the way you behave. Okay? These systems, and the way in which these systems are biologically implemented and the way the biological implementations go wrong in terms of the computations are central to understanding drug addiction. Neuromodulatory systems are central to almost every issue that you could name in psychiatry. And so, not so many years ago now, I guess 2006, we formed a unit in Houston, Texas. I used to be at Baylor College of Medicine and Rice University in Houston for 17 years, called the Computational Psychiatry Unit, built just on that instinct I gave you there with that little kind of sick illustration. And the idea was, yes, you can decompose the nervous system at all these scales, and we have to know that because that's the biology of the nervous system, and those are our knobs that we have to intervene or to understand or to listen in. But you could also reorganize that data or alternatively organize that data under computational ideas. And that's what one would do here. And what did I go to? Okay, this is the DSM-4 criteria for autism spectrum disorder. I didn't put it up for you to read, but if you peruse any of it, if you know anyone with autism or you have a family member that has autism or anywhere on the developmental disability disorder spectrum, you'll know this is a pretty good description. It's a pretty good description. It really fits. It would fit your experience watching a kid afflicted with autism. What it isn't is it's not a bunch of scientific dimensions. Okay? It's a really good description, but it doesn't act as a bunch of scientific dimensions. What you need in scientific dimensions are other ways in. And I think one of the things you could have to augment what we do in the area of psychiatry and the treatment of mental illness in terms of the nervous system is a kind of computational psychiatry. So there are units popping up all over the world. There's one in London. There's one being formed in Asia. The National Institute of Mental Health has a funding initiative along these lines. Let me just give you one example, and I'll be out with this here. So I went from squirting juice in people's mouths in functional magnetic resonance imaging machines that can eavesdrop safely on your brain activity, to running social exchange tasks with 2 people at a time. Okay? I borrowed games from the game theory world. I crafted them for my own ends. I set 2 people in an interaction. This particular game is called a trust game. One of the things that these games require that you do is think about the other person. What is that other person thinking? And given that they think that, what are they likely to do next? Okay? The trust game goes like this. This guy's called the investor or the proposer. They're endowed with $20. They can keep it round over. Or they can send any fraction of that $20 to the partner, and it earns a 300% return on the way over. So if I send 10, it triples to 30. Control passes to blue, and blue can keep it all or send me any fraction back. Suppose he sends 15 back. Well, I've made 5 on a risk of 10. Okay? Well, so what? Well, the so what is you can form a normative player here. You can make a mathematical rendering of the optimal way to play this game. You can then set real people in their interaction in this game, and you can use that mathematical model to decide whether or not they're playing optimally and whatnot. And it's quite a good probe. And it's a good probe for 2 reasons, and I've listed them here. One is this same little game here engages the same prediction systems that I was talking about earlier for just deciding whether or not this cue predicts you're going to get a drug or juice in your mouth. Okay? The same systems are engaged. They shift around according to this model I've shown you, and that's interesting. And they behaviorally parse traditionally defined psychopathology groups. By that I mean you can take a game like this, apply it to a group of people, let's say 1,000 people. Some of them have some psychopathology, like borderline personality disorder, major depression, autism spectrum disorder. A particular case would be autism, where one of the problems is a perturbed capacity to think about the other person, to put yourself in their shoes. Autism is a pretty large catch bag, so I don't want to just summarize it glibly like that. But any perturbation in your ability to think about the other person and to think about that other person's model of you perturbs this interaction. If you can't do those 2 things, you can't get a job. A job interview is basically image management. I want a job from you, and so I have to have a model of who you think I am, and this is who you think I am, but I want it over here. So I have to be able to send signals to you to move my estimate of your model over here. We call that second-order thought. I can look at you and see how you act. I can model that. And then I have to have some idea of your model of me. Otherwise, you can't do interactions like this. That's why we think this is a good game. I then had this extremely productive postdoc, Brooks King-Casas, who came to the lab and produced just an enormous number of papers and results and whatnot. I'm just going to summarize them quickly. The first thing he showed is you use a game like that, you do functional magnetic resonance imaging, and you see the sorts of shifts I was just showing you earlier. Okay? If this had been a light that came on here, this is time, and this is a response of this region of your brain here. This is called the caudate nucleus of your brain. This is where we were recording in human subjects before during the neurosurgery. And we kept doing the game, then this transfers back to this early point here. It's not transferring back on stimuli that are going on. It's transferring back based on the plans of the person into the future. So in other words, we sorted these signals according to what move this person's going to make 22 seconds into the future. So as they plan their actions into the future, you're engaging what looks like these same prediction error responses in your striatum that would be engaged if you were shining a light and following it with juice. The same systems that you lose in Parkinson's, the same systems that get addicted by every drug of abuse. In addition to that, he showed that there were neural responses that you might use to act as a biomarker for both borderline personality disorder and a separate one that you might use in an assay of autism spectrum disorder. I then acquired funding from the Wellcome Trust for Principal Research Fellowship from the Wellcome Trust to go after this in a big project between London and Roanoke, Virginia. I set up 19 sites in north central London. That took a couple of years, and we've been doing a variety of forms of these two-party exchanges where we record from one or both of the brains, and we're developing neural signatures in the context of these short games. They're a short game, 10 rounds. That's all it is. Here's the instinct of that, and then I'll finish on that. The instinct here is that healthy humans are sensitive detectors of interpersonal exchange. If there's something wrong with the way I'm interacting with you, even if I'm just sending you some money back and forth, and you're not quite fair, or you're wiggling around too much, or you get angry too quickly and the cooperation breaks down, you have a lot of hardware and a lot of software devoted to that. So we thought we could use healthy humans as kind of sensitive detectors of what was wrong with the people on the other side of the interaction. We used that instinct and developed a way to classify, we developed a Bayesian method to classify the trajectories through this investment repayment schedule. So that's investment 1, repayment 1, investment 2 for 10 rounds. And using that, we could separate different groups, cluster them easily. We could tell whether or not there was a borderline personality disorder patient on the other side of the interaction. We could tell whether or not it was a person with major depression on the other side of the interaction. We could tell whether it was somebody with autism on the other side of the interaction. Then we can start making computational agents that play in the style of those players and then characterizing those further mathematically. So that was quite inspiring. And it was inspiring to Pearl Chu, who was, Pearl and Brooks are both professors now, but Pearl was a postdoc from Harvard who was very interested in that. And just to summarize in one sentence what she found was, she developed a signature that this game engenders in the brain. That is a brain response that's uniquely elicited by this two-party exchange that is parametrically diminished to the degree to which you have autism. Okay? So it's a response in something called the cingulate cortex. That doesn't matter. This is the response of the cingulate cortex on the x-axis here. And this is the ADI rating. That's it's a caretaker rating of severity of autism. The more this response is gone, the more autistic you are, okay? According to that scale. We thought it was related to their model of themselves. And we spent 2 years developing that idea using picture assays where we showed kids pictures of themselves or pictures of other people, pictures of familiar objects, pictures of unfamiliar objects, pictures of favorite objects, and pictures of neutral objects. And we developed the idea that we could literally show them one picture of themselves, do a functional MRI for 18 seconds, and use that brain response to classify to the degree to which they had autism spectrum disorder. And that work is James Liu. He was an MD PhD student at Baylor College of Medicine. It doesn't matter the details of what the response looked like. And it remains to be seen whether or not this will work out. But this is the kind of thing that this two-party exchange engendered. So instead of a blood test, you might be able to take a kind of game test, establish a number, And that could be another quiver, another arrow in your quiver for thinking about what the disease state that they have. I'm going to end on breakout. This is breakout here. Breakout can't speak to you, but breakout can beat every human on this game where it has to move the thing around. It was trained on pixel data up to superhuman performance. And what I wanted to point out was the value function that they use. It's a little cryptic here. It's exactly the thing I showed you before. It's exactly the thing that's in the bee's head. It's exactly the thing that we think is in at least a subclass of dopamine neurons. These guys, this is from Google company called DeepMind. It's in London. That's been sort of wiping out the board game playing world. And then now they're trying to develop agents that develop senses of themselves. They're familiar with some of this work we've done. And I know for a fact they're looking for agent-specific programs that they can use to move into the medical domain. So thanks for having me tonight.
So thank you very much. Are there questions?
So you've snuck into my room on a randomized basis, and you've injected heroin. So I'm asleep and I never wake up. I don't wake up, therefore I don't feel any effects from it.
There's no sensory precursor to which you can assign the value. So when you inject heroin, your dopamine system sings, okay? And it says, go looking for anything that predicted that, okay? But you're asleep. I mean, it could be that you really start to want to go to sleep. Or you wake up going, I really enjoyed my dream last night.
But I don't know what it is.
You don't know how to assign it. You would never have the idea. I mean, if you're a normal person, you'd never have the idea, oh, I should go out and find some heroin today. I'm either going to do my potted plants or I'm going to go find some heroin. You just wouldn't, you know, my stomach hurts. You wouldn't, it's not something that would occur to you to do. You wouldn't reorganize your behavior around it. So these proxies get assigned the value, the artificially elevated value that the drug causes to be communicated to the rest of your brain. So it says this is more valuable than anything you've ever experienced in the natural world. You better learn about it. And then you generalize on that. It's not just the idea of the drug that does it, anything surrounding the drug, anything related to it. And also your nervous system generalizes. Once you've learned that kind of thing, it moves out into related things. People that have panic attacks, for example. You go to the mall, you have a panic attack in DSW. Is that a shoe place? DS, okay. I thought it was a tire place. I started thinking, what is a tire place doing in a mall? Okay, you have a panic attack there, okay? And if you don't get treated for that, it can egress. So the sensory cues that set off the panic attack start moving out, and they'll often move out to the curb of the mall, and you can chase you back into your house almost. It's that kind of learning. It's a learning problem. And it's complicated for lots of reasons. One of them is it's not like you're out of cognitive control either. You can think through things and whatnot. And these are drives. You feel that. But you would never want, you would know that heroin would make your stomach feel better.
I have a related question. Would you call you're physically dependent on heroin?
Sure.
Physically dependent.
Sure. But you wouldn't know that. Yes. So, I mean, suppose I put a little heroin release gadget in your gut. You know, I snuck in, so I'm having trouble breaking into your house every night, so I do surgery on you one night. I put a heroin release gadget in there and then let it release on a randomized schedule. You would completely dependent on heroin. You would just be sick. You'd be going to the doctor all the time.
Is this thing on? Oh, okay. I may have completely missed the point of what you were doing, but when you have the test for to show where on the spectrum somebody has the autism, does that help you find the cause of autism?
It could well do. It could do a variety of things. It could monitor the treatments that you have. You could say, here's where you are when you don't have this response, and here's what this treatment is doing to this brain response. The brain response is in specific areas that have connections in them. You could use that to inspire a series of experiments to go looking for what are these things connected to? What are those neurons doing? It's a human being, and so you have you can develop face validity over, it's very hard to tell if a mouse is autistic, okay? They don't have friends, they sit around. I mean, I don't know what an autistic mouse does, but the way they make autistic mice now is they say this mouse isn't doing normal social behavior. It has features of its behavior that seem to have some verisimilitude with human behavior. And that's not irrational, it's just, that's hard to check. So with the people, you don't even have to have a theory of autism. You can just say, if you're over here, we consider you on the spectrum and you can't go to school, and we could maybe use these responses to monitor something that moved you over there. So that's a biomarker. That was the idea. That is the idea.
Yeah, obviously you spoke about the heroin, but what about the abuse that's happening with prescription pills that kind of mimics...
with what pills?
Prescriptions. So you're getting prescribed these ADHD medications. Sometimes
oh yeah, you can definitely get hooked on...
Yeah, and people don't need them, and then there's abuse that's happening, and your dopamine levels are going up and going down, and it's a major problem, I think, within especially the younger generation. Is there research being done about the side effects of...
Oh yeah, there's lots of work. Not my work, but there's lots of work at many levels. So sort of, if you take Adderall, for example, in high school because you feel like you have to compete, do you later on develop drug dependencies at a higher rate than normal? The effects are very heterogeneous. We don't understand that very much. And I think giving kids methylphenidate is always a hard decision for parents. And I don't think we know the answer to that very well in the long term. There's a lot of interest in it, though, at the National Institute of Drug Abuse.
There's a question up there.
Hi. So there is a lot of discussion about dopamine. What about serotonin level? Have you ever, do you know anything about this disease?
Yes, I know a few things about it. I know 3 things about serotonin. I can spell it.
Perfect.
That's one. Well, as you know, one of the classic treatments for depression is selective serotonin reuptake inhibitors. So it slows or blocks serotonin reuptake. For any amount of serotonin released in your brain, it wafts around longer. However, one mystery there is it takes about 2 to 3 weeks to reach clinical efficacy. Okay, so the electrode that I showed you there, that we've developed, that we're hoping to move on to wires, will be a way to monitor both serotonin and dopamine simultaneously, and we've sped that method up 1,000-fold. In animal models, serotonin is actively investigated, but it's confusing. It's confusing. Ramalenko has left. Anyway, it's confusing. It's a mixed bag. It has an interaction with dopamine. When you get Parkinson's disease, for example, and you de-dopaminize your striatum, the serotonin terminals are known to sprout. Dopamine can be, if you take L-DOPA, dopamine can be transported into the serotonin terminals, so they come out. And serotonin is thought to be mainly related in the neurons that have been well classified to aversion, and patients, sort of active patients, learn to wait. Like if I was learning to do a task where I had to run across an electrified grid to get a piece of cheese, and a cue came on that said, hang on, it's electrified, I would have to learn to wait. Or maybe I would take a little punishment knowing that it's not going to get any harder and to get it, that sort of thing. But it's much more poorly understood.
I'm here. With your computational approach, how early can you diagnose people, for instance, for autism? How young can you already?
I don't know. That's a study of 25 high-functioning boys between 13 and 21, average IQ of 103. So it's a first pass. I think we'd have to get a population of 500 or 1,000 to know that. It's a pretty agnostic approach, though, but it's also not perfect. It's a statistical guess. There's a lot of work on this, though, so we're hopeful.
So as a very much a lay person, looking at people who are getting diagnosed or going through issues of mental illness, depression, there is no real way to diagnose. There's no medical way to take a blood test. But your work is pointing towards this.
Lots of work is pointing toward it, not just mine. Lots of work. I mean, in medicine, if you go in because you're turning yellow, or your head is killing you, or your elbow is swelling up,
Right.
yeah, they take your blood out and they do tests and stuff. If you go in and you say, I'm getting a little paranoid and I'm feeling this way, and they talk to you. And that frustrates every family that in here has had mental illness touch their family. I mean, we all have. That's very frustrating. So we like the idea, and young people in particular for computational psychiatry like the idea of working on basic science issues that might have an impact on mental illness and producing tests. Just producing a few little tests would be good. So that's what it's after. That's what computational psychiatry is after.
So you started out speaking of Tesla machine. They're in the parking outside. They're already there. We have them. So robotics is here. It's nowadays. So what is happening after? What do you expect? Where are we going? So it went very fast. You just summarized 30 years, 40 years of progress that led us to almost have a robot with a human brain, like a Tesla. Where are we heading?
Well, the robots are going to have superhuman brain. They're not going to have our brain. I mean, people, where are we going? I don't know, but I'll tell you this. This phone in my pocket, Samsung 5, which has its own hair growing on it. There was a computer when I was a postdoc so I started with Terry's quote, which, by the way, if anybody knows Terry Sinofsky, that was quite the rhetorical flourish. I don't know that I've ever heard him say that, speak like that. We were working on a Paragon, which was a parallel supercomputer. And this machine has more RAM in it, more memory, and it has faster CPU than the Paragon had. So what's funny is that in short 30 years, people used to make mice that ran around, hooked to the biggest computers in the world, watch it run its maze. The father of information theory, Claude Shannon, used to make artificial mice. And they'd run around these kind of crappy mazes. And they were like the size of a vacuum cleaner. And then they'd run around and they'd kind of keel over. And they'd say the fastest machines in the world are governing that mouse. Okay? And it was always kind of pitiful. What's interesting is the moment we have something that completely wipes us out in what we thought were strategic games, now it's not the way we think. We're not big lookup tables. And, well, maybe we are. And maybe that won't matter. So I think it's anybody's guess where it's going. It certainly has eliminated a lot of jobs. Automation has.
So on that note, artificial intelligence for the future, I want to thank Reid for a great lecture. I also want to thank the hosts for our dinner parties that some of the scientists are going to go to tonight. Thank you very much for doing this. It's a long tradition at Cold Spring Harbor Laboratory to have the community host dinner parties in their community, so thank you for doing that. For those that are going to the dinner parties, there are signs outside. If you don't know who you're going to, you can check out on the table. There will be a sign with the name of the host, and if you congregate around there, we'll arrange transportation to the dinner parties. Thank you very much, and thanks, Reid, once again.